Hi, everyone. Welcome. Happy New Year. Thank you for joining us for the webinar. We're really excited to have Dr. Nieva here. I participated in one of Dr. Nieva's studies with GO2 Foundation, and it's the Epidemiology of Young Lung Cancer study. And it's looking at, possibly how people who don't have the ordinary risk factors for lung cancer under the age of people under the age of 40 being diagnosed, what are the risk factors? What is what are they finding that possibly, whether it's in a medical history or an environmental risk factor or a genetic risk factor, family history. And so it's, if any of you on right now are were diagnosed under the age of 40, I highly recommend that you look into it. It was really easy. GO2 and Dr. Nieva make it really easy for you to give blood and answer a survey. So it's basically for people located in the US with non-small cell lung cancer. And I think it'd be really interesting looking at specifically small cohort of EGFR positive lung cancer. So again, if you're diagnosed under the age of 40, think about participating in this study, and we will provide a link for you. But what Dr. Nieva is here to do tonight is to give us a presentation on understanding the clinical trial process, which can be quite complicated. And so if you guys have questions throughout the webinar, please use either the chat or you can use the Q&A to type in your questions, and Ivy and I will collect them. And at the end of the webinar, Dr. Nieva will have an opportunity to answer some of the questions. So without further ado, Dr. Nieva.
Thank you so much, Jill, for that kind introduction. And thank you and Ivy for the opportunity to speak to this important group. Lung cancer advocacy is one of my favorite things in medicine, because advocates have now such a powerful role in guiding how we do research and in guiding what are the things that are important for us to study and look at. So I would like to encourage everyone who's on this webinar to really think of themselves as someone with an interest in lung cancer, someone who wants to advocate for lung cancer research, and really feel free to feel empowered to really guide what it is that we're doing, because everything that we do in academic medicine, we're doing for you. So let's talk a little bit today about clinical trials. I often get asked by lots of patients, hey, can I participate in the clinical trial, or isn't there a clinical trial for this? And I want to make sure that the people on the call kind of understand, what's this all about? What do clinical trials do? So we're going to talk today about the purpose of clinical trials in oncology. Why are we doing them? We're going to show you what a typical clinical trial portfolio looks like at a university. We're going to talk about who gets in and who gets out, who's included and who gets excluded. We're going to talk about how the results are disseminated, and then I'm going to talk to you a little bit about new tools that we're developing to make clinical trials a little bit more democratic so we can get them out to more people. And really, the reason we do clinical trials is we want to show that there's a better way of doing things than how we're doing it today. When I first was looking into being an oncologist, I remember an interview at my fellowship interview with Lou Slater at UC Irvine, and this would have been about 23 years ago. And a patient's X-ray had been brought into him, because that's how you did it 23 years ago. An X-ray was brought into the patient, to the doctor's office, and it showed a lung cancer tumor that was bigger than it was on the prior X-ray. And again, this was X-rays, not CTs. And the patient's tumor had grown, and I said, well, what are you going to do next? What are you going to do for second-line therapy for this patient? And his answer to me was, there is no second-line therapy for lung cancer. So to look at where we were 23 years ago and where we are today, the progress has just been fabulous. And that's all because people did clinical trials that showed that new drugs were better, new technology was better, or that using old drugs in a new way was better. You know, when we first had the EGFR inhibitors on the market, when Iressa came to market, everybody thought that the people who needed Iressa were people who had high levels of EGFR expression. And it wasn't until it was on the market for a few years that we learned that, well, no, actually, it's not the high expressors that matter. It's the patients with mutations that matter. And that led to one of the first revolutions in lung cancer, was recognizing that mutation was actually the important thing to look at and not expression. But we didn't know that the first few years we had these drugs. And this is, of course, why we do it. Lung cancer remains the #1 cause of cancer death in both men and women. And so we need to get better. And this is what the landscape looked like, you know, when I was in Lou Slater's office 23 years ago. We had 4 FDA-approved drugs for lung cancer. One was a nitrogen mustard derivative, one was anthracyclines. We had methotrexate, and then cisplatin was the big innovation in the 1980s. This is what the world looks like as of a few years ago. We now have multiple immunotherapy drugs. I made this slide last year. It's already out of date. There's another immunotherapy drug that's come to the market. Lots of targeted agents and lots of chemotherapy agents have been approved in the last few years. Immunotherapy, of course, has been, you know, the big miracle in lung cancer, but the EGFR patients have largely been left out because the current immunotherapy drugs that we have don't seem to have a big impact in the EGFR population. And I would love to have survival curves that have this flat part on it, where nobody's progressing. This flat area on the survival curve is really what we want to see, because that means long-term remission or maybe cure for some of these folks. And we always want to see this flat area as high as possible on these graphs. So we really need to study from the standpoint of the immune system. We really need to study the immune system more in people with EGFR mutations, because PD-L1 based checkpoint inhibitors like Keytruda probably aren't the way to harness it for EGFR patients, and it needs to be harnessed some other way. So clinical trials. We start doing cancer research in the laboratory. We discover things in cells and models, systems, and then we move drugs to the phase 1 part. So for phase 1 trials, we do lots of phase 1 trials, and most of them fail. Most of those clinical trials don't show a benefit that we should take to phase 2. We enroll relatively few numbers of patients in phase 1 trials. And this is the phase of research where we confirm that we have the right dose in humans. We then do phase 2 research, where we find out in what percentage of humans does it work. More of these trials are successful and go on to the next phase. And really, these phase 2 trials are all about comparing against a benchmark. So a typical phase 2 study will take, for example, patients with lung cancer who are on osimertinib or Tagrisso, and say, well, if we expect that for patients on Tagrisso, we expect that about 60% of the patients will see their tumor shrink. If we add another drug to it, we expect that it should be 70 or 80% to be worthwhile. And so we'll run a phase 2 trial, adding a drug to Tagrisso, hoping that we see response rates that are, 10 or 20% better than what we expect. But of course, we then have to move on to phase 3. Phase 3 is where the rubber meets the road. We enroll more patients in the trials, and we always compare to the standard of care. So, for example, if we were doing a study that was osimertinib or Tagrisso against Tagrisso plus something else, half the patients would get Tagrisso, half the patients would get Tagrisso plus the new drug. Now, the patients in phase 3 might get Tagrisso plus a placebo for the new drug, but it would, of course, be unethical to give them a placebo for Tagrisso. Tagrisso works. So we would never have a placebo in place of something that was proven. Whenever you hear about a clinical trial having a placebo or a sugar pill as part of the clinical trial design, what it's replacing is always the thing that's of unknown benefit. It's never replacing the thing that has known benefit. We would always want to have patients get the known effective drug. And then if phase 3 is successful, FDA approval follows. So the way the pharmaceutical industry describes this is that they make pharmaceuticals. Pharmaceuticals make profits. Profits allow us to do clinical trials and research, and that then generates approved pharmaceuticals. And the speed at which this wheel turns is the speed at which we generate more success. A lot of people don't like slides like this because it suggests that, some people think there's maybe too much profits in the pharmaceutical industries, or they have these types of concerns. But this seems to be the reality of the situation. What it means for the pharmaceutical companies, though, is they want to see successful clinical trials that turn into pharmaceuticals. This arm here, they want it to have the highest success rate possible because they want this profit over here. So they're very careful about how their clinical trials are designed. They're very careful about who they allow into the trials. They want to make sure that the people who are in the trials can show that their drug actually worked. Now, of course, there's been a big revolution in lung cancer in the last 20 years, and the big revolution has been that we recognize lung cancer is not one disease. Lung cancer, although it affects 200,000 or so Americans a year, they don't all have the same lung cancer. They have lung cancers that are based on different molecular drivers. And for this group, the EGFR driver is the one of most interest, but it's not the most common one. The most common one is actually KRAS. For people with squamous tumors, they get FGFR1, and then we have all these other groups. And there's advocacy organizations available for a lot of these other groups. But the EGFR community is a very large and important one. What this means, though, is for a typical university that's developing clinical trials, we would actually have to treat each of these as a separate disease and have clinical trials kind of for each population separately. And this means that we have to maintain a large menu. Now, just like it's hard for restaurants to maintain a large menu because they have to have all these different ingredients and all these different items, for a university, maintaining a large menu of clinical trials means that we have a lot of regulatory effort. There's huge amounts of paperwork involved in clinical trial. There's a lot of government oversight and regulatory oversight. So the first thing to recognize is we don't have a clinical trial for every single situation. We have clinical trials for the situations that are common, and we develop clinical trials and we open clinical trials at our institution that we think can can enroll patients. So if you look, for example, at a menu that we have right now for EGFR mutated patients, we have clinical trials testing bevacizumab, lazertinib, patritumab deruxtecan. We have for people with metastatic disease, but I don't have any neoadjuvant or adjuvant trials for patients with EGFR mutations. And the big reason for that is that Tagrisso has recently shown benefit in this group right here. So everybody now who gets surgery with stage 1B disease and beyond should probably get osimertinib, and there are no competitors in this space. So there's very little research going on into new drugs here. Typically, the order of events is that we take people who have metastatic disease, develop drugs in metastatic disease, and then we move them to earlier lines of treatment. For people with BRAF mutated tumors, there is a clinical trial giving these drugs before surgery, and we have this trial for people who have more advanced disease. We have some small cell cancer trials. Alectinib has been around for a long time for people with ALK rearrangements. So we don't actually have very many clinical trials in this space. And it's actually very good news. We don't have clinical trials in this space because there are 7 FDA-approved drugs in this space. So there's not a lot of drug development going on because we actually, fortunately, have lots of great therapies at the moment for these patients. We have something for the NTRK patients. We have something for the high PD-L1 patients after surgery. We have something for the high PD-L1 patients in stage 4. Squamous cell patients, we have something. For people who have none of the above biomarkers, we have something else. People with HER2 mutations, we have poziotinib. And the latest biomarker on the street, KEAP1-NERF, we now have telaglenastat and sapanisertib. So what this tells you is that if you have lung cancer and let's say an EGFR mutation, and the center that you're at doesn't have a trial for you, another center may have a trial for you in that specific situation. And all centers maintain a menu. Some of us have bigger menus than others, but nobody has every item on the menu. You can't find restaurants that sell Greek food and Indian food. It's just too hard to do that. So we have to be somewhat limited. Now, there are, of course, a lot of regulatory safeguards for every one of these trials. Before we can open a clinical trial at USC, we have about 7 different committees that are involved in that trial. And each one of those committees is comprised of different people. So there are a lot of different eyeballs, lots of different brains that look at a clinical trial before it opens. The process at USC begins with scientific review. So if I designed a clinical trial myself, or if someone from the pharmaceutical industry hands me a protocol, or somebody from a large cooperative group like the National Cancer Institute or Southwest Oncology Group hands me a protocol, I have to turn it in for scientific review, and a group of scientists and oncologists will review it to make sure that it's scientifically sound, that the statistical analysis looks like it's going to be done correctly. Once the study has approval of a scientific review committee, the next step is that it goes to something called an institutional review board. These are ethicists, and these are people who make sure that the study's design is ethical. So make sure that we're not giving placebos to people who shouldn't be getting placebos. They make sure that the consent form that the patients read actually describes all the risks accurately. They make sure that the investigator doesn't have conflicts of interest that would impact how they would describe the study to others. And they then, if they deem that the study is done ethically, we can go ahead and go to the next step. The next step is radiation safety. With lots of clinical trials, we check frequent CAT scans. Sometimes patients will need to get a CAT scan or a PET scan or some other imaging modality, or maybe a biopsy. Well, all those things involve additional radiation. And so the Radiation Safety Committee makes sure that the amount of radiation that we're presenting to the patients for participating in the trial is safe and it's appropriate. If the study involves biologics, a biologic would be things like a modified virus that fights cancer. We do that every once in a while, or any kind of gene therapy in any way. We have a biosafety committee that reviews the protocol. So for example, right now for patients who have head and neck cancer that's caused by the human papillomavirus, we inject the patient with a different virus that creates an autoimmune response against the human papillomavirus DNA. Well, they want to make sure that that viral product that we're injecting into people is safe, and it's not going to cause a pandemic, and it's not going to make people sick. So before you inject anything that's biologically active into somebody, a molecular biologist has to look at it. Then the business people get involved, and they get a Medicare coverage analysis. Make sure that the things that we're doing on the clinical trial that are going to be covered by Medicare or by insurance are actually standard of care. We can't bill your insurance company for a procedure on a clinical trial that's considered not standard of care. So for example, if we're injecting you with a new drug that's not FDA approved, you can't bill for that. We're not going to bill your insurance company for that. The cost of that has to be borne by the sponsor of the clinical trial, usually a drug company or a grant or a foundation or a university. Similarly, if the clinical trial wants to do an extra biopsy, but an extra biopsy wouldn't normally be needed, well, you can't be billed for that. The study has to pay for the cost of the biopsy. We've been doing some work recently using tumor-infiltrating lymphocytes. And these patients who receive this treatment get hospitalized, and sometimes get hospitalized up to 2 weeks. All that's considered not standard of care. So the pharmaceutical company that's sponsoring the trial gets some of the largest hospital bills you can imagine whenever we enroll a patient into the study, because it can't go to insurance. After the patients are on the trial and they start generating data, we have a data and safety monitoring committee that looks at the data that's coming out of the study to make sure that there's no abnormal safety signals. I'm actually the chairman of the Data and Safety Monitoring Committee at USC, and I get a note to read every time a patient who's on a clinical trial lands in the hospital, or has a major complication, or has anything serious happen to them. I get alerted to it, and then we have committee meetings where we review all of those things on a quarterly basis and make sure that we're not seeing an abnormal safety signal where we should be stopping the study if it looks like there's a safety problem. We also have a data quality committee that reviews that the data that's being collected by the investigator and by the investigator study team is actually of good quality, so that the science that comes out of the study is actually going to be good. So all this has to happen for the study to work. And these are the ethical principles that guide us when we're designing clinical trials. The principles are beneficence. Beneficence is the idea that by participating in the trial, it's going to be helpful for the patient. We can't do clinical trials that we think are not going to at least do something good for the patient, or the patient's going to feel good about themselves because they're doing something for the community and it's not hurting them. We always have to think that there's going to be good for the patient and good for the community. Autonomy. Patients get to make the decision about clinical trials. Nobody's ever forced onto a clinical trial. Everybody can participate or not participate. Respect for persons means that we allow that person to make a decision in an informed way. We treat people with respect when they're on a clinical trial, making sure that they can drop out anytime they want. ask all the questions they want, understand everything they need to understand about the study before moving forward. And then justice. The people who take risks by participating in studies should be the population of people that benefit. So we ask EGFR patients to be the ones who try out new drugs for EGFR lung cancer. We don't try them out on random people that aren't going to benefit. And we try to make sure that everybody has access to clinical trials. So for example, at USC, we have the Norris Comprehensive Cancer Center where we see people with really good insurance. And then we have LA County USC Medical Center where we see people who have no insurance. And we try to keep the clinical trials open at both sites so that both the rich and the poor can participate and have that same opportunity. Now, once we open the clinical trial, they have very specific inclusion and exclusion criteria. Like I said, the pharmaceutical company wants their trial to be successful. So they want to make sure that the people who enroll aren't going to have problems unrelated to the cancer or unrelated to the drug. So, for example, a typical inclusion criteria for a study will be that, well, we're only going to enroll people with stage 4 lung cancer. We're only going to enroll adults. We'll enroll people who've had 1 treatment, but not more than 2. So they have to have had 1 or 2 prior treatments. If they've had 5 prior treatments, we don't want them. And the reason that the studies are designed that way is because they want to look at patients who have a very narrow expectation of how their disease is going to go. They want to look at the narrowest group of people possible because that's most likely to generate homogeneous results and a change in a homogeneous population. They'll oftentimes say that patients must have had a particular drug. Like for example, for an EGFR trial, there may be a requirement that the patient's previously been treated with Tagrisso. That'll be a common requirement because Tagrisso is so standard. The other thing they'll insist is that the patients have a good performance status. That means that the people have to be relatively healthy. They have to have measurable disease. And that means that you have to have tumor that's big enough that we can see the size of the tumor change in response to the treatment. So they'll want to, for example, allow patients who have a 3-centimeter tumor in their lung, but not a 5-millimeter tumor in the lung, because a 5-millimeter tumor doesn't change. It's hard to see the change in tumor size with that. They want to see that the drug that's administered in the trial actually makes tumor shrink. And this can be tricky sometimes because tumors don't shrink very well in certain locations, or at least it's hard to see the shrinkage. Like for example, bone. Tumors in bone don't change in size very much because the bone kind of molds to their shape. And then when you cure the tumor, the bone kind of stays distorted. It's kind of like a handprint in concrete. It doesn't change when the hand gets taken away. And so people with bone-only disease oftentimes can't participate in studies. There'll also be a list of exclusion criteria. And basically, these are things that could cause unrelated problems and could make the results of the trial confusing. So people with HIV, other organ problems, cardiac problems, blood clotting problems, other cancers typically can't participate. This is some information on performance status. I said people have to be healthy. If you're wondering how the doctor tells if you're healthy enough for a clinical trial, you could look at this 5-point scale from performance status 0, which means you're normal, to a performance status of 4, which means you're bedridden, and then everything in between. But most of the time what we do is we just watch you get on the exam table. So if you're wondering why it is that the doctor has you get on the exam table, but then doesn't actually seem like they're paying attention when they're listening to your heart or lungs, or they're chatting while they're doing that, we get a lot of information by watching somebody make these movements. You'll probably notice that your doctor never turns their back while you're getting on the exam table and starts looking at the computer. They actually watch how you do this. And so whether or not you're considered good clinical trial material or not good clinical trial material is a lot dependent on whether or not you hop up on the table or whether you struggle to make that movement. And it makes a huge difference. For example, this is data from a clinical trial that happened in 1994, where we looked at people who were performance status 0, so basically normal, healthy condition, versus someone who has to spend some of the time in bed, someone who's not quite as strong. And it makes a huge difference. The likelihood of the tumor shrinking in this trial went from 23% to 14%. The time it took the cancer to grow through the chemotherapy went from 4.3 months all the way down to 1.5 months if you included people that weren't healthy. The average survival in this study, and this was before targeted agents and before immunotherapy, the average survival in the study, if you were healthy, was 10.8 months, and if you were not healthy, it was under 4 months. And the likelihood of surviving 1 year was cut in half. So what this means for the pharmaceutical companies is if they allow more people with performance status 2 in their trial, the average results are going to be worse. And it's going to be much harder for their drugs to show a benefit to that population. So they're always looking for people who are going to be in the healthiest condition possible to test whether the drug works. And it's kind of tricky because the doctor and the patient don't always agree on what the performance status is. Most of the time, the doctors overestimate the performance status compared to where the patient thinks they are. Doctor and the patient actually agree less than 50% of the time. And most of the time, we assign them a 1 category higher, sometimes 2 categories higher. So think about what that means, 2 categories higher. This means that, you could be, the doctor may say, I think you're minimally symptomatic from your cancer, but you could be in bed greater than 50% of the time. So we're doing some research to try to make this more standard. It's one of the things I focus on, because studies have shown that if you and the doctor agree on the performance status, you actually live on average about 16% longer than if you're in any of the other categories. Women particularly get their performance status overestimated because women are actually very good at making themselves look good. They'll put on their makeup, they'll put on a nice dress, they'll come to the doctor's office looking good. And so the doctor says, hey, they look great today. And then they'll score them a high performance status. Men will show up in their bathrobe and slippers when they feel a little bit uncomfortable. And then they tend to get scored lower on average than women. Performance status is actually more predictive than age for determining how someone's going to do with a lung cancer diagnosis. If you look at this Pronopal score, which incorporates performance status, a blood test called the albumin test, another blood test called the LDH test, and how many metastatic sites the patients have, the most powerful predictor is the performance status. And you can see that the survival probability for people who are in good shape, a score under 3, is very different than A. So you can imagine when a pharmaceutical company looks at data like this and says, well, gee whiz, if we accidentally put a higher percentage of A-type patients who get our experimental drug, and the people who are on placebo are all in group C, the placebo will look better than our drug, just because we didn't properly make sure that our population was homogeneous. Same is true for safety. If you look at people who have more side effects from drug, the less healthy you are, the more side effects you have. And so when companies want to know whether or not their drug is going to have a lot of side effects or not a lot of side effects, it really depends on the health of the people that they put in their trials. So when they put really healthy people in their trials, the reports look like the drug is going to be very safe on average, compared to if they enroll very unhealthy people in the trial, then the drugs look unsafe because the unhealthy people have more problems that are just unrelated to the drug, but can be related to the cancer. So this is why it's very important to try to, when you're going through cancer treatments, to try to maintain a very high level of global health, because really the healthier you are overall, the fewer problems you tend to have. This is an experiment that we did where we put Fitbits on patients, or this was a Microsoft Band study. And we looked at how much time the patient spent not being sedentary. How many hours in a 60-day period were they up and active, doing not, marathon running or anything like that, just active, cooking, cleaning, doing household activities, gardening, and those sorts of things. And what we showed was the likelihood of you having a problem was much higher if you weren't active. These were all different ways of you having a problem. And the more active you were, the fewer problems you actually had. So a lot of the research I focus on is on using things like wearable devices to assess health. And this is really awful data from the state of California. In the state of California, if you take of 17,000 patients treated for lung cancer, and this is data from about a decade ago, about a third of patients get no treatment at all. And the big reason is because by the time they came in to see us, they were too frail. And all the data that I've shown you earlier on the impact of frailty on clinical trial design, it also means that all the drugs that are approved have shown to have benefit in people who are not frail, which means that there are no drugs that have shown benefit in frail people. And as a result, frail people oftentimes get no therapy. So we do a clinical trial and it works. What do we do after that? Well, we publish it in conferences and journals, and we get FDA drug approvals, and it gets incorporated into society guidelines. It gets insurance coverage. It goes out on Steve Liu's Twitter page. So all these things happen when a clinical trial works, and this is all good. So when you see these things, that's usually a very good sign that something good has happened in the treatment of the disease. We're actively working at USC to try to bring clinical trials to more people. At USC, we're located, right here, but all these little blue dots are community cancer sites of HMO patients. And we've found that HMO patients oftentimes can't participate in clinical trials because they're restricted to where they can get their care. And they oftentimes can't come to a university. So we've reached out to these groups, and we've actually created a program where patients who are in an HMO can be screened for clinical trials at USC. And if they're eligible, they can come in and they can participate. And this is something we'd like to see more universities doing, and reaching out to this group of patients that I think are a very important group of patients that oftentimes don't have the opportunity to participate in. And this is part of making sure that we do clinical trials in a way that incorporates justice as an important ethical value, that we want everyone to be able to participate. So for that program, we use telemedicine. And this is the way telemedicine looks by doctors who still have their hair. The patient can be there, you can share the screen and share the scans. And we now consent patients to clinical trials using telemedicine. We're also opening up a study where we actually use data from wearables during the telemedicine visit, because telemedicine is a little bit like a webinar. None of you know whether or not I can walk because you can't see my legs and can't watch me get on an exam table. The same is true with telemedicine. We can't watch the patient get on an examination table. So we need other sources of data, such as what we can get from wearables, to try to figure out whether that person is healthy and active and able to be active and eating well and doing all those important things during the visit. So currently we're doing this study with Genentech. This is going to be for people getting treated with a subcutaneous formulation of atezolizumab, where we're going to actually do all the treatment at home. They're going to get a clinical trial where we're testing not whether or not the drug works, because we already know this drug works for some lung cancer patients, but we're seeing if the drug can be safely administered at home. So using a subcutaneous pump delivered at home by a home nurse, using wearables to monitor the patient. So this is an example of a clinical trial that's testing not a new drug, but a new technology and a new approach. And then, of course, as Jill mentioned, we're participating in the Epidemiology of Young Lung Cancer study to really understand lung cancer in younger people and why it's occurring. So I don't know if many of you followed the news on this, but in the year 2020 was the first year that we started to see publications that for the first time, women had more lung cancer than men if they were diagnosed at a young age. And so what's going on with that? Well, here's some data from the United States on tobacco use. And tobacco use peaked in the mid-1980s in the United States and has been dropping ever since. And it's been dropping for men and women about equally. So we now see relatively little tobacco use. And now this has been great for men. The incidence of lung cancer in men has been dropping in a manner that's very parallel to what's gone on with tobacco use. But what the heck's going on with women? Women stopped smoking around here. And so the curve should be going down here. And it didn't. It actually kept going up. So one of the things we're trying to understand in this Epidemiology of Young Lung Cancer study, and this, by the way, with lung cancer in women, this is very often occurring in younger women. This is often occurring in people with EGFR mutations. And we know that EGFR mutations aren't tobacco driven. So what's going on? We want to answer that question. So we're trying to look at people who were diagnosed with lung cancer before the age of 40. We're looking for people like the EGFR resistors that we know have lung cancer frequently at a younger age on average than other subtypes of lung cancer. If you participate in the study, you take a survey and we send someone to your house to do a blood draw. The questions that we ask are things about demographics, medical history, lifestyle, environmental, genetic risks, and then there's a maternal history that we can get for people whose mother is able to take the survey as well. It's only for the people in the United States. We're not able to do this overseas because that makes the environmental exposures a little more diverse than what our epidemiologists can handle. It's anybody with non-small cell lung cancer before the age of 40. And we do require that everyone participating in the study have done biomarker testing. So if people want to participate, they can sign up online at go2foundation.org/younglung. I think there's a few other links in the chat that people can use. If you yourself weren't diagnosed before the age of 40, but you know someone who was, and they've not participated in the study, please share the link with them. This is something that we really do need help with. You don't have to come to USC. The study's entirely online, and we would love to have you participate. So with that, I would be delighted to answer any questions. I do see there was one question from Yan Ling Fan about copies of the slides, and I believe this whole session has been recorded, and Jill and Ivy will provide a link to where you can look at the recording at your convenience, or you can share it with people who may have interest in clinical trials.
Yes, we will. It'll be available both through the EGFR Resistors newsletter and in our Facebook group. There's actually one other question also that I see that was posted that says, which committee that reviews trials has lung cancer advocates on it?
We don't have any specific advocates that are disease-specific reviewing clinical trials. Now, we do at the IRB, the ethics committee level, we do have community members present, and every IRB has community membership requirement. But there may not be a lung cancer advocate on that committee. It may be a lymphoma advocate. or a breast cancer advocate. And in general, the IRBs are always looking for people who want to engage. And so if you're someone who wants to engage with clinical trial review, I would encourage you to contact your local academic medical center. There will be a place for the institutional review board, and reach out to them and say, hey, I'd like to be involved. And again, they really are always looking for people, particularly anybody who can commit to some time, because you do have to read very long documents and then go to long meetings.
Yeah, and if we can talk about that, some research advocacy stuff at a later date as well. So you can reach out to Ivy or myself if you're interested about in doing research advocacy. Dr. Nieva, there's another question in the Q&A. Somebody who said, I've read that Latinx make up only 3.4% of clinical trial participants. Why is that?
Frankly, because we haven't done a good job of getting clinical trials to that population. So one of the things that we do at USC is we make sure that the LA County population, which has a much higher percentage of Latino patients, and the Norris Cancer Center does, make sure that our trials are open there. So that's first and foremost. But the LA County Department of Health Services, the governmental organization that runs that hospital, doesn't really want to promote clinical trials among the population because it takes more work. They get more infusions, they get more scans. They wonder whether or not that's a good investment of the county resources. So it's not necessarily promoted in that group. The other thing you'll find is that underserved populations, whether they're Latino populations, African American populations, oftentimes they're not getting their care at academic medical centers, which is where the majority of clinical trial enrollment occurs. They're getting their medical care in community centers, and oftentimes those community centers don't offer clinical trials, much like the HMO networks in Los Angeles County don't, and they have a larger percentage of Latino populations as well. There's additional barriers of trust. Some people and some populations and groups don't trust clinical trials as much as others. Particularly in the African American community, there's been a very sad history of things like the Tuskegee experiments and a number of historically bad ethics that were carried out in the name of cancer research, and that has caused distrust with certain communities, and that trust is going to take a long time to earn back. I think there's a lot of different reasons why we don't get as much participation from those groups as others.
Here's another question that has just come in. How can a person who is outside the US participate in a clinical trial?
Well, with airline tickets primarily. The challenge for people that are outside of the United States is, there may be clinical trials open locally. So, many of the clinical trials are worldwide global trials. So, when, Genentech opens their next big trial of subcutaneous atezolizumab, it's going to be opened by their global oncology group. So they'll have sites in France, Spain, and Germany. There'll be sites in Eastern Europe. There'll be some sites in Asia. So there are clinical trials internationally. Those do exist. But I read the question to mean, how can I participate in a US-based clinical trial if I'm international? So there's barriers. The barriers relate to, usually you have to be around. Most of the therapies are, administered intravenously. So you have to be here every 3 weeks, or even if it's an oral therapy for a clinical trial, you have to check in once a month, and you got to check in once a month in person for most clinical trials. I have an interest in making clinical trials more democratic and more available through telemedicine. But I think we've got a ways to go before that becomes routine and standard. So most clinical trials, you still got to show up, you still got to be in person. So it takes a lot of airline tickets back and forth from where you are to the trial site. I would plan on 1 to 2 times a month flying in to attend a study related visit. And if you miss too many visits, you do get kicked off. So you do have to be compliant.
It looks like we have time for one last question. Here it is. Many patients are intimidated by the medical world. When asked to participate in a clinical trial, they may have trouble refusing. What incentives do researchers have to encourage patients and sign them up?
So the big incentive is that most of us believe in these trials. And studies have shown that patients who participate in clinical trials on average do better than people who don't. So the big incentive is we want what's right for our patients. We want them to do well. Now, when we open a clinical trial, we expect that we're going to enroll patients to it. Now, in some places, there's economic incentives for the physicians to enroll in clinical trials, and maybe private practices will have that type of structure. Most universities do not. So most people who work at universities, whether they enroll 1 patient in a clinical trial or 50 patients in a clinical trial, they don't make any more money. So there's not a sort of this direct monetary type incentive. There is an expectation that if you work in academic medicine, you'll participate in clinical trials and you'll enroll patients in clinical trials, and people think you're not doing your job if you're not advancing the science, and we advance the science by participating in clinical trials. So I would imagine that if you don't enroll patients, at some point your boss will look at you and say, "Hey, what are you doing? Why aren't you advancing the science?" So I think there is that incentive, but I would say that for someone who's intimidated by doctors and who always wants to do what the doctor tells them to do, I would bring another set of ears, and I would bring another set of ears from somebody who's a little bit pushy. We all have somebody in our life that's a little bit pushy, and that's a great person to bring to your doctor's appointments, or if you can't bring them in because of COVID, have them on the phone next to you on FaceTime saying, "Hey, what are your incentives to enroll, and really what's this trial all about?" Make sure that you feel comfortable, and no good academic oncologist is going to try to twist your arm to enroll in a trial. If we don't actually want you on the trial, if we have to coerce you, because clinical trials are a big pain in the rear. If you don't really want to do it, there's extra doctor's visits. There's sometimes extra biopsies. You have to show up and do these visits and take these surveys and all these sorts of things. So if you don't want to do those extra things, and you're someone who's going to be complaining about those sorts of things, and don't have good buy-in that it's what you want to do, it's not going to work well. It's not going to be a good relationship on the study. So yeah, only participate in a study where you're like, yeah, I want to do this, and I want to do this, and I understand it. And we all, as oncologists, if our patients don't understand the trial, we don't feel good about it, because really we're asking you to participate in a research project. You need to understand what you're doing, because there's going to be some unknown risks and some unknown things can happen, and you have to know what you're signing up for.
Great. Thank you so much, Dr. Nieva. I think we are just at the time for the end of our webinar. So on behalf of the EGFR resistors, we'd just like to make sure that you know how much we appreciate you spending time with us today and answering our questions about clinical trials and talking to us about the epidemiology of young lung cancer study with GO2. There's information in the chat with links to that study, and you can also find that information in our EGFR resistors group as well. Again, if it applies to you and you were diagnosed under 40, it's a really easy study to become part of, as Jill mentioned. And again, thank you to all of the members of the EGFR resistors and anyone else impacted by EGFR lung cancer who logged on today to be part of this webinar. We appreciate all of you. And if it wasn't, for all of you, we wouldn't be able to make an impact. We all need each other to work together. So thanks again, everyone.
Thank you, Jill. Thank you, Ivy. It's really my privilege to speak to the group, and feel free to reach out to me if anybody has any follow-up questions by email.
Great. Yes. Thank you.
Definitely.
Thank you so much. And thank you to GO2 for hosting the webinar for us. So
Yes.
we appreciate you providing this platform and helping.